a professor from Graduate Institute of Food Science and Technology from National Taiwan University. And it's my honor to be here to talk about some food safety control and quality evaluation, especially in food microbial contamination, spoilage, and fermentation. And I think I just briefly introduce myself. And I actually graduated from Furen University, Catholic University, majored in nutrition. However, then I also did my master and PhD as a nutrition. So I'm a nutritionist. But after I graduated from my PhD, I moved to and after I completed my PhD research, I switched my research field into yeast genetics. So part of my training actually is in yeast as a yeast geneticist. Okay, so then I moved back to Taiwan. Then I came into the Food Science Institute. Therefore, I started my bacterial work, microbial work a little bit, and then I extended my yeast research into some other bacteria and also in food safety. And so today I would like to, as you can see from the slide, my subtitle is the food microbial contamination, spoilage, and fermentation. Since this is a really huge field, or huge content. So I'm trying to give a broad view of microbiome, especially you will see my outlines. I divided into 4 major parts. The first one will be the landmark discovery in microbiology. And the second part will be the microorganisms in food. I think these 2 parts you all may be already familiar with it. However, I would like to put much more efforts in the past and current status of microbiology. And in this part, I will try to use different examples, and mostly that would be my own experience or the research that I have been working on in my lab to do the food fermentation and food spoilage and food pathogens. Okay. And the last part, I think the last part maybe overlap with other professors in other lectures a little bit. So I think I will mostly focus on the first 3 parts. Okay, so I think when we talk about microbiology, we always know that the size of microorganism is always an issue. So in terms of microorganisms, then which also it means that the size of the microorganisms cannot be seen by naked eyes. So you will see from an animal cells, the size of the animal cells is approximately about 10 micrometer. Okay. And for yeast, it's about, for bacteria, it's only 1/10 of animal cells. And so bacteria, it's probably approximately around 1 micrometer. And in between of bacteria and animal cells, mostly will be the single yeast cells. So the single yeast cells, that size is around, in the middle, it's around 5 micrometer. Okay. And then you can see that virus and small molecules that would be much smaller than the bacteria, which that can be seen even by light microscope. So you have to use the electron microscope, then you can observe these microbes. And for the basic types of microbes, including bacterial, fungi, parasites, and prions and virus. And here you will see parasites it also be discussed in some of the food safety issue. However, I won't talk about any parasites in today's talk. And the prions actually is protein. And however, the protein may infect it from different animals. For example, the most famous prion is to cause the mad cow disease. So that can transmit it from one cow to another cow. Okay, so although it's not live microorganisms, however, when we talk about microorganisms, the new idea is included the prions in it. But we won't talk about this either in this class, okay? And the virus we know, especially like the COVID-19, everyone is afraid of that virus. However, for the traditional food science, we won't include the virus as a food pathogen, right? You all have the food science background. However, we now, I think most of the countries started to include virus in the, especially for food safety issue, and especially for the, what's the virus name? Norovirus, rotavirus. And because the norovirus or rotavirus, it's spread it by, it's transmitted by your saliva or from your hand, etc. Although it cannot grow in food matrix, but it can pass by humans through the food material. So now, if we concern the food safety issue, we always count the rotavirus, this virus in food safety related to the virus infection. Okay. So for the science part of the microbiology, I always like to, I know most of you may already know this famous scientist, but I still want to talk about them because you can, from these history, you will know the first microbiology is really a key issue when we discover all these events through the history. And first you will see from Louis Pasteur, and you will see that we all know that he's the one to discover or to develop the sterilization, and also we call it pasteurization. It's because the Pasteur, his last name is Pasteur, so we call this way, the way we sterilize our food or our other facilities, that we call it pasteurization. That's by the name of Louis Pasteur. And you will see that the first time he discovered this food microbiology actually is through the fermentation. And because he's a microbiologist, and he's well known that he knew that the fermentation is caused by the growth of microorganisms, especially when he worked in Lyon, in Paris, and as a scientist in the winery. And this is a small, that was a small winery, and because some of the wineries in that Paris, nearby Paris, they found that sometimes they have the wine as too sour. And sometimes if you get a good wine, it's not sour at all. So by discover that Louis Pasteur is the first person to take a look the microbial communities under the microscope. And at that time, he found that in a good wine, he found there's a microbiomes which is round shape. However, in the soured wine, he found that the microbiome is elongated. It's not round. It's rod shape, ROD, rod shape. So that's the first time to distinguish a microbiome growth is related to the wine quality. Okay, so the good wine should have the round cells, and the sour wine will have the raw taste. So that's how we started to know, and by using heat treatment, and the rod shape microbiome can be killed. So that's why it started to develop the pastoral sterilization method. And he's the one who noticed that the round cells that can convert sugar into ethanol, as we know, that's yeast thereafter. So the second person actually is very important as well. You can see from 1905, he actually developed the technique for growing bacterial, especially for cholera and tuberculosis, and which made him to win the Nobel Prize. And why I say he had a great contribution in food safety, because he developed the first, we called it core principles, which means if you want to identify food safety or food toxication or any food safety issue, you need to identify a specific bacteria. And the specific bacteria need to be inoculated back to the original food matrix and showing the same phenotype as the contaminated food. So that's the Professor Koch had the great contribution on food safety. And as you can see, why he can get the Nobel Prize by isolating the cholera and tuberculosis. That is because for the past, especially in the early times of the microbiology development, you will see most of the, it's difficult to get the pure culture, okay? And as you can see, in our food matrix, we always have a lot of microbiomes. So that makes the identification or purification really hard at that time. So Dr. Koch used a method to trying to isolate the bacteria. And the way he did, for the past, most of the time we use the liquid culture. And started from Koch, he tried some solid culture. And the solid culture, for the first time he used the solid food as a culture medium. Can you think what is the first solid medium for the cell culture, for the microbial culture? Meat. I mean, you need to separate isolate the culture. So for Koch, he was he used potato. He cooked or steamed the potato or cooked the potato, and then once the potato was cooked, he sliced in the middle of the potato. Okay, so when you have a round-shaped potato, you slice it, and then you got two flat surface. And then he trying to they're trying to identify or isolate the cells on the surface of the potato. Okay, and then he found it's not quite you can imagine that potato is not nutrient-rich medium. So then he was thinking, trying to use other materials to isolate the bacteria. However, when you are doing mediums, the mediums need to be solidified. And how can you solidify a potato? Because potato, you know, it's hard, it's solid. But for a medium, how can you solidify the medium? We all know that we use Petri dish, and we have the agar plate on the Petri dish. It's quite easy. But at that time, they did not know how to make the solidified matrix. Okay, so because he has an assistant, his assistant, he had an assistant, and the assistant was very interested in taking photographs. And at that point of time, we had to develop the films by hand. Okay, it's not by machine. So by hand, then you have to develop the photographs against the gelatin. So the gelatin is one of the materials that they had to use to develop their films. So they made the medium with the gelatin in there. And in that case, the gelatin can be solidified, and then you can isolate your strands from the gelatin plate. But there's a disadvantage by using gelatin, because if you know that gelatin is, the melting point for gelatin is really low. It's about 25 degrees. So at this point, if you are limited at 25 degrees Celsius, and then you won't be able to identify or isolate some pathogens, because most of the pathogens, they will survive at 37 or even higher temperature. So they were thinking to using agar plate instead of the gelatin plate. So the agar plate actually can tolerate much higher temperature, so it won't melt at 25, even won't melt at 37. In that case, they can isolate some pathogens. And by using this method, that Dr. Koh developed the techniques growing bacteria, and especially the cholera and tuberculosis. And that's why he won the Nobel Prize in 1905. Okay, so that's very interesting, because every time I talk about this to my student, they didn't know the history. But we always know that we have the agar plate. And I think most of us won't think about the original story, how the agar plate was developed. Okay. And the other one is the Dr. Hansen. You can see that is he is a Danish mycologist and fermentation physiologist. Why I put his photo here, because you can see in 1883, he was the first one to isolate pure yeast culture to produce beer. And because I'm an yeast geneticist, so I pay a lot of attention about research related to yeast. And at that time, you can see Dr. Hansen, actually, he worked in Carlsberg company in Denmark. I'm not sure whether you know Carlsberg. It's a very famous beer company. Okay, and he worked there as a scientist. And he was the first person to isolate a pure yeast and also apply to the beer industry. So for most of you, when you drink the beer from the commercial beer, mostly that would be pure yeast culture. Okay. And I will talk about that later. And there's another person also won the Nobel Prize in 1907. He's Dr. Buchner. And he is the scientist, he was the scientist that had a great idea which had much more impact on our microbiology fermentation research, because at that time we always thought that yeast, for example, the wine was made by the yeast fermentation, and the fermentation, how the fermentation happens, we never thought about it. We thought that's because of the yeast itself, or I'm not sure whether how you think about the yeast. But from Dr. Buchner, actually he had the idea about the cell-free fermentation. What is cell-free fermentation? Which means that's cell-free. When you do the fermentation, there's no cells in there. From the surface of the words, you know the cell-free fermentation. However, the real idea is he did the experiments that he cultured the yeast, and then he collect the yeast cells, and then he grind it. He dry the yeast cells and grind it, and trying to destroy all the yeast cells, kill all the yeast cells, and then put the yeast cells back to the medium. And do you think it still can do fermentation? Yes? Yes. So that's why he called it cell-free fermentation. Okay. So why is that? It's because the yeast cells, they can secrete proteins into the medium. So actually the fermentation is the enzymes that yeast produced. However, the protein was secreted into the medium. So the cell-free idea, cell-free fermentation idea, that actually is the yeast cells secrete proteins into the environment in order to ferment sugars. So this is this was a quite new idea at that time. I think even now some students still don't know what's the process of the fermentation. Actually, it's through the protein digestion, through the protein function to cause the medium, the components in the medium change. So we call it cell-free fermentation. That's why you see that he got the Nobel Prize in Chemistry at 1907. Okay. So as I said, this cell-free idea, cell-free fermentation, actually had a great impact on the synthetic biology happened in current. So let's come back to the microorganisms in food. And for all the microorganisms in food, generally we can describe them into 4 categories. And the first one is the fermentation microbes. And as I said, for the most of the research for the past 100 or more than 100 years, even 1,000 years, always fermentation was first discovered through people's life. Okay, so you can see the fermentation microbes, the function of the fermentation microbes actually is to improve our food quality. And however, there's through this fermentation process, sometimes you may get contaminated microbes. So you will have the spoilage microbes, and the spoilage microbe which will cause the spoilage of food. And so that's the food safety issue. And again, also with the pathogenic microbe, sometimes it's difficult to distinguish these 2 spoilage microbes and pathogenic microbes. However, you will know the pathogenic microbes will cause the health hazard. Okay, so this is the pathogenic microbes. And the last one, it's interesting too. And we said there are a lot of inert microbes, and which means we don't know what these microbes do in the food matrix. Okay, but it's really do nothing? I'm not sure. Actually, I think through a lot of new techniques, people trying to understand whether these inert microbes are actually doing something, but just we don't know. Okay, So I think the inner microbes you will find it's always in the environment, it's always in your food matrix, but we don't know what they are doing currently. Okay. Okay, and then the next part is the one that I would like to explain a lot and trying to use a lot of examples to trying to explain the past and current status of the food microbiology. And hopefully, at least the example I gave you are those that I'm interested in, and also my current study may focus on those parts. However, I think there are more and more to discover. Okay. So first, we talk about the past food fermentation. We all know that, we always know that from wine to vinegar. Okay. So if you want to, and you want to produce wine, however, it becomes vinegar. They still have a lot of cases in our food industry when the company want to produce wine, however, eventually they produce vinegar. So that means there's some food microbial contamination or other problems. But for the past, people are trying to understand the wine and vinegar, and also the microbes associated with the wine and vinegar. And of course, we know the alcohol and Saccharomyces. And I will have another slides to introduce you the alcohol and Saccharomyces. And then even now, we still are interested about the lactic acid bacteria and yeast fermentation, especially for those probiotics study. And the fourth is the pure yeast and beer fermentation. That's, as I said, that from Dr. Hansen or from Louis Pasteur, it's always discussed the pure yeast and the beer fermentation. And for the current status about food fermentation, there's key issues in this research area, including, as I said, the probiotics and the function of probiotics. And even now, we are trying to understand the relationship between probiotics and prebiotics. Okay, so that's one of the research focus. And the second one will be more technical issue. That's the gene sequence of probiotics. And because there are so many probiotics, and you really want to make sure, especially now, a lot of company or research, they are focused on the gut microbiota. When you isolate specific bacteria from gut microbiota or from food safety issue, you isolate this bacteria, you need to have the gene sequence for those bacteria. Okay, so even for the probiotics, we still need to confirm the gene sequence. And the third part is the bioengineering of microbiology. That's as a lot of company trying to use bioengineering to engineer the microbes to be a specific strain to do some probiotics or produce some active bioactive compounds, etc. And the fourth part will be the effective production or preservation of starter cultures. For example, if you have the probiotics in your culture, and however, when you want to make your probiotics into a product, then you need to have a specific way to preserve your cultures in order to get enough live cells. And you know all the production process they may through the heat treatment or other treatment, and that will kill your cells and reduce the cell numbers. So that's why to get the effective production or the preservation of your starter cultures is really important in food industry. Okay. And for now, what's the food fermentation what's the really interesting question in the food fermentation at the current state? Let's look at the yeast first. And because, yeast is a unicellular, and as I said, it's a single cells, but it's a eukaryotics. So it's a round shape or rock shape as well. And it's larger than bacterial, and as I said, it's in between it's around 5 micrometer. And you will see there's a list of yeast cells here in this slides. And most of them were exist in the fermentation system, okay? And however, somehow, like the Debromyces and Hansanusa or the Tauropus, and exist in the fermentation medium, such as wine or yogurt. And however, they may cause the sour taste in the wine or in the yogurt. So, but it's not spoilage. It's not. It's a spoilage, but not pathogenic. Okay. Except the Candida. Most of the Candida, then it would be pathogenic yeast. Okay. And Pichia is the one who used the most in the bioengineering for the yeast. Okay. And for the past, we know the yeast in food, and we can use that especially in the bakery product, because it can ferment sugars to produce carbon dioxide, and the gas can give the porous texture in the bakery products. So and it's also leavened the bread. So yeast can be used, or it's very commonly produced every day in our bakery industry. And it also contributes to a flavor by formation of alcohol, aldehyde, and esters. So that's the metabolites of the yeast. It gives the product with the good flavors and good textures. And the yeast fermentation usually happens in beer, wine, vinegar, or some pickled vegetables. And now, and I think a lot of people are interested about the beer. So I use beer as an example. And as you can see, I'm not sure, maybe you like beer or not, maybe not. But I'm not sure how many people know ale and lager. Does anybody know the differences between ale and lager? Okay, so some people said top fermentation and bottom fermentation. And which is top, which is bottom?
Lager is bottom.
Okay, right. Lager is from the bottom fermentation. But for most of people, you may just, all this beer. They have different color. They have different flavor. And they have different smells. Okay? All right. And what's the top fermentation and the bottom fermentation will make difference? How can they make difference? And why they make differences? Aerobic and anaerobic. But they are yeast. Okay. So let's see. For the past, we know that so for lager and ale, generally we can use this cartoon, this picture here. Although the actual color may really different by different ways of produce or get from your wheat, or how long you roasted your wheat. That may make differences. But the basic differences for ale and lager is due to the ale is mostly, if you are in Europe, especially in England, because I studied I was in Scotland for quite a long time. And we know that for most of people, especially for Britons, they will go to pubs during the weekend and trying to get a beer, a cup of beer. And what types of beer they are drinking? I think they prefer the dark beer and with the, as you said, the top fermentation beer. Okay, so when you drink those beer, the color is it's brown color. Okay, and the yeast may suspend it in the medium, not in the medium, in the beer. Okay, and for lager, that is the beer that you get most from our commercial company. So when you go to the supermarket, you buy the beer, mostly will be lager beer. So the lager beer, actually, it looks different from ales because it's light colors, mostly. And it smells different because ale beer actually smells, they always have strong smell. Okay, so that's the major differences. And when you taste those beers, you will know the ale beer, they have also strong taste. It tastes very differently. And the lager actually is a light beer, so it tastes much lighter than the ale. Okay, so from the past, we know the beer, you can look, or you can know the beer by their looks, by their smells, by their taste. And you can distinguish the major two different types of beers. Okay. And what's the current study? The current study is we are not only looking at the beer color, beer smell, and beer taste. We are looking at what type of yeast were fermented in ale, and what types of yeast were fermented in lager. Okay, so actually the difference is for ale, it's fermented by Saccharomyces cerevisiae. However, for lager, it was fermented by Saccharomyces petroneus, and it has an old name called Saccharomyces carspergenesis. Why is Saccharomyces carspergenesis? Because the carspergenesis is, do you remember we said that Dr. Hansen was in Carlsberg company, and he isolated the first purified yeast to do the beer. Okay. So the differences is between these two strains. And more interestingly, you know, I will talk about that later. But there's another difference is the optimal growth condition. And one of the top fermented yeast, we called it Saccharomyces cerevisiae, which fermented to produce ale. And it was a warm fermentation, which means the fermentation temperature is a little higher compared to the Saccharomyces petroneus for the lager beer. And the yeast to ferment the lager beer, which always use like 15 Celsius. However, for the ale, it may use like 20 or 25 Celsius. So the temperature that for these two yeast are different. Okay. And then there's more differences if you look at the chromosomes by using the genome sequence data for the current study. And it shows very interesting. It shows that the Saccharomyces cerevisiae to produce ale lager actually is the diploid cells. I'm not sure whether you know the diploid cells. Okay. Diploid means the cell, actually it's very interesting for yeast. actually there's sex in yeast. If you look at yeast cells, they have the MET-A mating loci, which is A cell, calls it as an A cell. And yeast cell also have alpha cells. So when you mate the A cell and alpha cell together, it becomes diploid cells. So it has each chromosome, they have 2 sets of chromosomes. Do you understand that? Like for human, we have 23 sets of chromosomes, and then we have the total number is 46 chromosomes, right? For yeast, if a single haploid cells, they have 16 chromosomes. And so if they are diploid cells, then they will have 32 chromosomes. Okay? And then you look at the Pastronius, the Saccharomyces pastronius. They are allotetraploid, which means they have 4 chromosomes for each set. Okay, so it was caused by a diploid cell combined with another diploid cells. So normally if we said single cell, they have 1 set of chromosome, and diploid cells, they have 2 sets of chromosomes. But for these larger strain, this Saccharomyces pastronius, they have 4 chromosomes per set. Okay? Hard to imagine? Okay, we will see the cartoon later. So for a lot of research, then we know there are a lot of changes of these yeast cells. Actually, is from the environment. So that's why the differences for, the characteristics of the beer actually is really rely on the cells which fermented the beer. Okay, so the fermented to produce ale and lager beer. So that's totally different. Okay, so how does that happen? It's actually studied in 2011. It was discovered very interesting because when we do the wine or lager or actually all kinds of fermentation, you know the fermentation is happened not in the natural environment. The fermentation actually is from a brewing environment. It's handmade environment. Do you agree? Right? For yeast, if that's in the natural, the yeast always grow on the surface of fruit, the surface of some vegetable, and also in probably in soil. However, in a fermentation environment, you can see what's the typical condition in that fermentation environment. Huh? No oxygen? What else? Nutrients. Nutrients. What kind of nutrients in the fermentation medium? What is the most important nutrient? Sugar. So for the fermentation, you need to put high concentration of sugar, right? And then the sugar will become ethanol, alcohol. So then it's from high concentration of sugar turn into high concentration of alcohol. So that's not the natural environment. So we called it as a domestication or domesticated environment, 驯化. Okay, so it was in a particular environment which provide the yeast with high sugar, and then it turned into high alcohol. And actually for a natural condition with high concentration actually give the cells with high osmolarity, right? 渗透压, the high osmolarity at the beginning of the fermentation. And at the end of the fermentation, it was in a high alcohol concentration. So that gives the cells with a lot of stress as well. So that's why in these two very extreme conditions, which would not happen in the natural selection. So in this wine or beer production environment, it was found that the brewing environment may cause the domestication of the yeast cells. And then you will see there are two types. If you're thinking from this graphic, you will see the light green actually is the natural environment. So the natural environment, actually, it was found there's Saccharomyces cerevisiae. And it also found there's another strain called Saccharomyces eubayanus. Okay, and then once it get into the brewing environment, there's accident happens. But the accident actually was directed by the domestication. So what happens? It happens that you can see from the color. The Saccharomyces cerevisiae were in the brewing environment, but we know before the pure, the techniques for isolate the pure strain doing the pure culture, there's some environmental strains will come into this environment as well. So by accident, the Saccharomyces eubayanus, they combine with the Saccharomyces cerevisiae. So that's why you see that's the diploid cells. This is the diploid cells. And then when they combine, they become tetraploidy cells. And the tetraploidy cells that we now here call this is the early Saccharomyces pasteuronius. Okay. And then the early Saccharomyces pasteuronius was found turned into modern Saccharomyces pasteuronius. Why we called it modern? Because it was modified by DNA recombination, and which is my my field for the past. It's very interesting that you see, for example, you see they should have two yellow color and two orange color. And originally they have the blue color. So originally that's 1:1 ratio. I mean, in terms of the chromosome, it's 1:1 ratio, okay? And then it become to, you will see the yellow color is still keep most of the yellow color. However, the blue one turned into the red one. And it was found the blue one actually coded with SAL1 gene. The SAL1 means sulfur. Sulfur. Sulfur? Liu. Sulfur. Okay. Encoding the gene which can produce sulfur dioxide. And sulfur, not sulfur dioxide. And so that's why we hypothesize the original Saccharomyces pasteuronius with strong flavor, which is from the air or from the eubayanus. They all have this kind of the gene. However, in the modern pasteuronius cells, the sulfur gene was inactivated. So that's why when you taste the lager beer, now you can feel that it's not as strong as ale. Because in ale, they still keep the sulfur gene. However, in the lager, the sulfur gene was inactivated. Okay. So that's quite interesting. And not only the sulfur gene was converted, and also were inactivated. it also have some genotypes that changes, and phenotype changes, not genotypes. Of course, it's also genotype. You see the length of the one of the chromosome from Saccharomyces cerevisiae becomes shorter. Okay, so which means I won't go through all the details in this strand, but if you are interested, you can discuss with me, or you can take a look. But this paper is quite difficult. Okay. So actually, we found that through the domestication, the Saccharomyces cerevisiae were turned into Saccharomyces pestronius, and which may change the phenotypes of the beer, okay, including the flavor, the taste, and the color, okay. And not only this, because in the brewing environment, you will see, because as I said, the cells in the brewing environment, it has to tolerate the high concentration of sugar, high concentration of ethanol. So eventually, these cells will die. When the cells dies, they will release their DNA. And although most of the DNA will be degraded in the medium really quick, however, they still have chances. It's just the opportunity, the frequency. They still have the DNA can get into another cells. Okay, so you will see that when these pestronius cells were degraded or they will die in the medium, these Saccharomyces pestronius lysed cells can release their DNA, and the DNA will come into still in the environment. However, when in this environment they have other yeast, they will contaminate it into other yeast cells. So that's why when you see that's one of the chromosome originated from the modern pestronius come into the original Saccharomyces eubayanus, then it will turn into other Saccharomyces baianus. So that's the domestication process from the original Saccharomyces cerevisiae and Saccharomyces eubayanus, and then turn into pestronius, and then it also get involved into these contaminated yeast in this environment. So they will have to produce new strains, and these new strains, for the past, we won't know which one is the new strain, which one is responsible for ale or for the lager. However, by genome sequence technique, we can know the processing and what happens during this domestication environment. Okay, so that's quite interesting, especially, I think this is a big discovery for the past few years. And since then, it resolved the yeast mystery for 100 years. Okay. So next, I think we just finished this Aspergillus oryzae, and then we'll take a break. And for the fermentation in the past, we all know, I think this, although I cannot drink, but I think this wine, it must be, it's a kind of famous sake. If our friends in Japan, I think you all know this wine. And it was fermented by Aspergillus oryzae. Okay, we know it's really good, so it has been registered for Japanese Aspergillus. Okay, so however, we know there are so many types of Aspergillus, so many strains of Aspergillus, and some of them can produce yellow pigment, some cannot. Okay, and one of the and this is the structure of the Aspergillus under microscope. What's the interesting part for the Aspergillus oryzae in the past. And we know the first isolated Aspergillus oryzae is in 1876, actually by a German scientist. And he isolated the strain from Koch material fermented by the Aspergillus oryzae in solid state cultivation. However, I'm not too sure which metrics that, which kind of koji that he isolated. But when we talk about koji, it includes fermented soybeans for making soy sauce, or bean paste, or that can sacrifice the rice and other grains, or even potato in making of alcohol beverage, such as sake. Okay. And for the past, we know the Aspergillus, it's a microbes or fungus. It grows really rapidly, and the mycelium grows and into the rice kernel. And as far as I know, that when we want to make a high quality sake wine, you have to remove the surface protein of the rice, and then to produce a really pure starch part for the rice kernel. And then you can get a really good sake. However, you need to have a really good strain as well. So the sake wine will give you a pleasant fragrance and accumulation of flavoring compounds. And again, through 2000 to 2015, I mean, it's around the same period of time. From, I think, through 2003 and 2015, in between of this period of time, because of the development of genome sequencing, there's a lot of new, interesting research have been shown that the Aspergillus oryzae, you can see they have, although it's a higher eukaryotics compared to yeast, but they have less chromosome. They only have 8 chromosomes, but they have like 12,000 predicted genes. Okay, so this may not mean anything to you, but you know that Aspergillus oryzae seem to have evolved by domestication. Again, it's in the wine and the sake production domestication. But it's really interesting that it's involved by the domestication. However, we thought the domestication is happened from Aspergillus flavus. And Aspergillus flavus actually is the strain which can produce aflatoxin and yellow pigment. So for safety issue, of course, we don't want to, because the sake is using, sake was produced by sacrifice the starch, the grains, okay? Different grains. So if you are using the Aspergillus flavans, then it will contain aflatoxin. But through this study, actually, I probably forgot to sign the, oh yes, the probably next page. You will see from the sake fermentation, the colony color and texture and the quality, they all, especially the production of the secondary metabolite is the aflatoxin. It's from Aspergillus flavus. However, through this fermentation, domestication, somehow the Aspergillus oryzae will not produce aflatoxin. Okay, but why? And you see this study, sorry, I still forgot to put the original journal paper citation on top. You will see it's really interesting. They compare the Aspergillus oryzae that makes sake with the Aspergillus flavus, and they compare that about 20 strains. And you will see what does that mean. And this is the relationship, the roots from this strain. Okay, that's another type of analysis to see their correlation between different strains. And you will see the green one, this is the Aspergillus flavums, this complete Aspergillus flavum genome sequence. And then you will see the blue starting from here. At that time, it's still called Aspergillus flavus. Okay? And gradually, they start to switch their genome into Aspergillus oryzae. That's very interesting. They found the different strains and different Aspergillus. Then they can separate into this cartoon, and you know the evolution. The domestication actually is a part of evolution. And the evolution is from the green and gradually showing up the blue, and then it becomes blue, the processing. Okay. And interestingly, from all these strands, they pick up 4 strands, and 3 of them are the Aspergillus oryzae to make the sake, and another 3 Aspergillus flavus. And you can see this is the gene expression profile. And what is the gene expression profile? It's the gene to produce aflatoxin. And the black color means the lower as the minimum and the maximum. So it turned yellow color means they can produce a lot of aflatoxin. And when you produce, it's all, when the result showing all black, which means they have no or very little of aflatoxin. Okay, so through this genome database and this genome sequence, we can know more in detail about these different strains. Okay, and which can explain why the Aspergillus oryzae, although this I only show the aflatoxin production, because this is the key. Because this is the key, because you won't, you cannot use the Aspergillus that can produce aflatoxin to produce sake. So you have to get rid of the aflatoxin genes, no matter it's deleted or it was inactivated. Okay, so this is the story of the Aspergillus oryzae. Okay, let's take a break and we'll continue later on. And any question you want to ask, you are free to ask. next part. Okay, so this is Aspergillus oryzae. And then we come to the lactic acid bacteria. Okay, so the lactic acid bacteria are a group of microorganisms that are normally used as starter cultures in the manufacture of a large variety of fermented food. And I know most of you are familiar with the Lactobacillus, Lactococcus, and Bifidobacterium. Okay. And I think the idea about the lactic acid bacteria actually is originated from the scientist from Russia. Actually, in 1910, Professor Mechnikov, he proposed that without any science evidence, but he proposed that aging is caused by toxic bacteria in the gut, and that lactic acid can prolong life. So he wrote a book called The Prolongation of Life. And actually, he took the yogurt every day, just support his own theory. And then I think he died about 89, or 79, almost 80 years old. At that time, it's pretty long life. Okay, so Professor Mechnikov actually is an immunologist. So he studied the phagocyte. Okay, and he got the Nobel Prize by the immunology study. However, he always thought from the infection idea, he thought the potential life-lengthening properties of lactic acid bacteria, and that's why he drank the yogurt. He drank the yogurt fermented by Lactobacillus bulgaris. And now, at that time, it's only from bulgarix. Now, for most of the yogurt, it was fermented by the Lactobacillus bulgarix and Streptococcus thermophilus. And that's the major lactic acid bacteria in the yogurt. And however, at that time, it was not popular to drink yogurt at that time. And the key person who promote the yogurt drink the most is the Japanese, we call Shirota, Mr. Shirota. He invented the yogurt-like probiotic drinks containing the Lactobacillus casei strain, Shirota. And so from that time, it's in 1935, I think the students in Japan may familiar with this drink. We all in Taiwan are familiar with drink as well. And I think the inventor Shirota also investigating a causal relationship between bacteria and good intestinal health, which eventually led to the world marketing of kefir and other fermented milk drinks or probiotics. And so we all know the fermentation by lactic acid bacteria. But for now, how we think about the lactic acid bacteria, we know that lactic acid bacteria is now, we call it grass bacteria, and because it's generally regarded as safe. And however, there's some study showing that the lactic acid bacteria also inhibits the mucous membranes of the intestinal and reproductive tracts of humans and animals. And incorporation of selected strain into certain fermented food has gained increasing interest in recent years. That's what some of you are interested, because now, no matter in the company industry or in the research field, people are trying to, or scientists are trying to isolate it or selected strains to ferment food, and trying to get the probiotics and prebiotics and as a product to provide as a supplement and trying to increase the health effect for human. Okay. And now currently there are some challenges encountered by the lactobacillus during travel through the GI tract, our gastrointestinal tract. I think most of you may be aware of this. However, you will see why is that? So actually the point to trying to develop such a lactic acid bacteria, you need to optimize the bacterial. Why is that? Because the bacteria, once it get into our mouth and esophagus and go to the stomach, you would know the stomach is with low pH. And then it turned into the small intestine. There's 3 parts of the small intestine. And the intestine, they will raise the pH around 7. So from pH 2 to pH 7, and then it comes to the cecum or the colon part. So for a lot of studies, they have shown that, I remember one of the speakers in my department a couple of years ago, he was doing the gut microbiota, and he said that according to their experiment, if you have your stew and you dry out all the water, the moisture part, then they analyze the component or the total amounts of the bacteria. They're about 1/5. 1/5. 1/5. The weight, they may weight 1/5 of the stool are bacteria. So you know there's millions of bacteria, millions and millions of bacteria in our stool. And from the, we said from the stomach, there's low pH with a lot of enzymes. And in the small intestine, there's also a lot of enzyme, especially in the duodenum. So you will see when they count the microorganisms through the passage of our GI tract, you will see there's only lower than 10 minus, lower than 10 fourths, that's 10,000 microbes per gram of food in the stomach. And then gradually the number will comes back. So in duodenum, it's about 100 to 10,000. And the end of the small intestine, that would be about 10 million, more than 10 million cells. And at the column, you will see they hypothesized that if you want to have the bacteria reach to your column, then initially you have to give them 10 to 12 cells. Okay, I should say, I'll say it clearly. If you want to have the cell remain in your stomach, then you need to have around approximately about 10,000 cells. Okay, then they can reach to the stomach. And if you have like 10,000 cells, it may pass through the duodenum, the first part of the small intestine. So if you want to have a product that contain your probiotics, and you want those probiotics can stay in the column, you need to have a large amount of probiotics, like 10 to 12 cells, then that can reach to your column part. So that's a lot of cells. And so for commercial product, you need to know the probiotics you have is not only correct probiotics, you also need to have enough probiotics. And also these probiotics can stay until their column. So mostly you have to characterize your probiotics, whether they can tolerate with pH 2, and also then it can tolerate at the pH 7 throughout to the column. So that's one of the key point. And also we said that, sorry, I forgot, where is that? Okay, so there's another key part that we can see from next slides. This is another example. And this example is the human milk glycans and microbiota in neonatal mucosa. And why I use this slide, because actually there's a very interesting study done in Africa. And the scientist is from Georgia University in United States. And he collaborated with the WHO and trying to study the microbiota, the effect of microbiota on nutrition status in African child. Okay. In that study, I didn't show the result. There are very interesting studies showing that in Africa, they took the stool from the baby at birth, and every month they collect the stool. And then they're trying to analyze the microbiota from their stool until they are 3 years old. So that's a huge project from WHO. And very interestingly, they found for the children who who suffer nutritional problem, they often shows the microbiota dysbiosis. Dysbiosis, which means they are not balanced. Okay, so which means the microbiota in infant, actually, they have a process called maturation. The maturation happens at 3 months old for those fetus, for those babies. Okay, so what's the signature for the maturation of microbiota? And that's very interestingly, they found at 3-month-old babies, they start to see in bifidobacterium to spike. Okay, so you can imagine where these microbiomes from for those babies. For a baby, supposedly they should be germ-free in fetus, right? Although I know there's some research talk about that even in uterus, they probably have some bacteria in the uterus. However, I think most of people still put a question mark on that. But we know when baby was born, for most, especially in Africa, through the vagina in their mom, from their mom. So they may receive the microbiomes from their mom, okay? But they also analyze the microbiota from mom and also from their babies. And they found the microbiota maturation actually started from the 3rd month after they were born. And during that time, the lactobacillus, especially the bifidobacterium, was spiked up. And so that's the signature. And they found when those babies with nutritional deficiency or malnutrition status, they do not see the spike for the bifidobacterium. So that's very interesting. However, the bifidobacterium, the maturation reached to about a year old, from 3 months to a year old. Then the bifidobacterium numbers still start to drop. So in the adult, if we look at all the microbiota data, you seldomly found bifidobacterium were isolated. Or even they were isolated, it's with very little amounts compared to other bacteria. Okay, so that's the one thing. And so then people start to think about what is the best food for these babies or for old babies. And then there's a group of people trying to study the human milk glycans. And also there's a microbiota study in neonatal mucosa. And it's very interesting. I think this is, this data is from a review data. I didn't go back to the original data. Okay, and from this data you will see this is our gut mucosa. So think about this is our epithelial cells in the column. And these with circle or triangle, there's many types of oligosaccharides or human milk glycans were drawn in these cartoon. As currently we know there are at least 30 types in this review paper, at least over 30 types of oligosaccharides in human milk. And supposedly these human milk oligosaccharides, they when the babies or have the human milk, they ingest of the microbes and these human milk glycans as well, because they suck the milk from mother. So the microbes actually come together through the suction. So they get the microbes from mother's skin, okay? So these milk glycans once come through because it can be digested, okay? It won't be hydrolyzed by these oligosaccharides as prebiotics, as natural prebiotics. It won't be hydrolyzed by enzymes. So these oligosaccharides will come through to the column. And interestingly, we think currently, I think most of people was convinced that these human milk glycans, they will attach to the surface, the mucus of the intestine, especially at the colon part. And then there's a study talking about bifidobacterium. They have a specific affinity to these glycans. So these bifidobacterium will have receptors to recognize these glycans on the surface of the mucus. Then this has become a selection medium. So the selection medium is select for the bifidobacterium. They can easily bind to this mucus. And in that case, it also help them to proliferate. Okay, so once it proliferate, then there's a competition between the pathogens and the bifidobacterium. So once, however, because we said that bifidobacterium has a specific affinity to this milk oligosaccharide, so it can be a key microorganisms in this environment. And then you will see that with other pathogenic, for example, the bifidobacterium here, we use green color, and the light green is the bacterials and the E. coli and the Clostridiums. Those pathogenic bacterials number will be reduced. So that's the microbiota in the neonatal mucus. And we also know if there's no these milk glycans, then the bifidobacterium, in other way, will be less in bifidobacterium will retain in the mucus, on the mucus. In that case, there's little bifidobacterium, but will be higher pathogenic bacteria in the gut mucus, the mucus of the epithelial cells, and which will these with these pathogenic bacteria that will cause the inflammation, and the inflammation will break down the walls in between the two mucous cells, epithelial cells. So that causes, we call it leaky gut. Leaky, L-E-A-K-Y. We call it leaky gut. And the leaky gut, then that will cause the inflammation, and also the inflammation, other inflammation signal proteins will come into the cells and will get into the body. Okay, so that's the milk, human milk glycans and microbiota in neonatal mucosa, and this is through the microbiota sequencing, and then we can know the different composition of these microbiota in the stools. And through all this study, then we can know this mechanism. So that's why the human milk glycans, actually we think that's still the best food for the baby. So that's why as a nutritionist, we always encourage the female mom better to feed their babies at the first at least 6 months with the milk, with mom's milk, which can have enriched glycans in the milk, which will help the babies to establish their gut microbiota as early as possible. Okay, so that's the current status for this prebiotics and probiotics study, and I think that's really interesting. However, it's also really complicated because even with the oligosaccharide analysis, it's a tough job to distinguish those oligosaccharides. Okay. Okay, then next part we will talk about the fermentation, about microbiome biotechnology. And this idea also is original. Actually, this is the idea. I draw this picture actually is because I think a few years ago, the professor from Tsukuba University came to Taiwan, and I'm trying to introduce him and summarize his talk in our college. And then because I'm doing some of this study research as well, so that's why you can see the biotechnology actually. For a lot of microbiology study, we can isolate the microbiome from natural environment. And now we know there's a lot of genome sequence data which you can get from website. A lot of online information you can just get, go into the database and search whatever you want. Okay, so that's quite available currently. And through here, we know there's some genetic manipulation technique, and we have cloning, overexpression, or even deletion techniques that can make you to create specific proteins. And the proteins can, through the protein structure study and enzyme study, or you also can do metabolomic study with the chemistry study. And now we are trying to combine all these techniques and trying to set up a cell factory, which is, in other words, we can say there's so-called synthetic biology or some cell factory engineering. So what is the cell factory idea? The cell factory idea actually is originated from 2015 as the first paper published in Science. It was rated as the top 5 techniques were developed in that year in Science Journal. But it's not about this. It's about the opium, yapian, because they can produce one of the components from the same as opium by yeast. Okay, so the bioengineering yeast. This is an interesting paper, and I forgot where it published, but it's much more recently. The scientists trying to make hoppy beer without hops. What does that mean? They make hoppy beer without hops, which means they're trying to make beer by yeast itself, but they don't put hops in there. And why is that? Because they identify the major component from hop, and they know the flavor is come from specific components of the hops. So they express the hop genes into yeast. So when they culture the yeast, the yeast can produce the hops flavor, and without fermentate the hops itself. Okay, so that's the idea about the bioengineering, or we can call it synthetic biology. Why is synthetic? Because you synthesize the gene in the yeast, not by natural, it's by human cloning, by human to clone those genes into the yeast. Okay. So why can you do that? Or this is it's also from my student. We actually haven't completed this experiment. And so we're trying to bioengineer the yeast, and what we are interested in is the mogoside. It's 罗汉果, mogoside extract. There's a specific compound, we call it mogoside, and those mogosides are natural sweetener. And we have done this for many years because we know 罗汉果, the monk fruit, it has been limited to China because it has a great value. So the China government prohibit that export of the mogosides. So the export of the monk fruit, 罗汉果, they cannot export the fresh fruit, but they can export the dry fruit, because you cannot do anything from the dry fruit. Okay. So we're trying to make these mogosides from yeast. And there are a lot of companies in Europe. They're also trying to make steroid or other flavonoid or flavone compounds through yeast, and as a bioactive material or drugs. Okay. And how can they do that? Because we all know that for animals produce cholesterol. So for plants, they produce plant sterol. Okay. And for mogoside, all those triterpene drugs or flavonoids or the ergosterol, those plant sterols, they have the same pathway at the very beginning. So in this case, you see, we want to synthesize the mogo or mogosides from the yeast. However, the mogosides actually is from acetyl-CoA and become HMG-CoA and become mevalonate. Are you familiar with pathways? Huh? TCA cycle. Yeah. It's from the energy production, the acetyl-CoA from lipid degradation. And then you have to go through synthesis. And because yeast, they have on their cell membrane, they need to have ergosterol, it's kind of plant sterol, to maintain their structure of the cell membrane. And also maintain their maintain their osmobility. So for these eukaryotic cells, especially for yeast or fungus, they have this process to produce ergosterol. And to form the ergosterol in the cells, you will see that's from acetyl-CoA, HMG-CoA, and through mevalonate. And there's an enzyme called HMG-CoA reductase. Actually, this is the enzyme, the key step enzyme, rate-limiting steps enzyme for ergosterol synthesis and also for cholesterol synthesis in animal. Okay. And then from mevalonate, they have this so-called mevalonate pathway. And then they have to go through a steroid pathway. And then from steroid, they will have another pathway to go through the triterpene pathways. So in my experience from our students in my lab, they're trying to change the gene expression from this HMG1 and this ERG9 and ERG1, trying to get this particular compound, we called it 2,3, this is squalene, and the squalene is the combination of those triterpene terpene compounds. And then from the squalene, they started to cycle, they have to become with a lot of cycle chain. And so there's enzymes to produce squalene and also 2,3-oxidosqualene and become the ergosterol. Okay, you don't have to remember all these. I just give you the example. So we are trying to overexpress the enzyme over here and trying to get a lot of precursors, and then they can go through here. Then I think this is currently, so far we know, this is the last available component in yeast. And then we have to introduce 3 different genes from this mogosai fruit. And then we hopefully we can, one day we can produce the mogosai 5. In that case, I don't need to actually buy the fruit. I don't need to extract from those fruit. I just need to get it from my culture. And if I can do it, then we can get the mogosai from the culture and from the cells. And then we can get the natural sweetener that can reduce the requirement from environment or from time or from weather, from climate, etc. And so this is the so-called synthetic biology study. Okay, so I think we finally finished the fermentation work. And now we are talking about the microbes and for the spoilage. And the spoilage, you will see that microbial spoilage actually is the growth of spoilage microbes and their active metabolism of food components. And also the spoilage microbes, usually they will release a lot of enzymes that will react with changed properties of food component leading to the spoilage. Okay, so it's very often we've seen that for the past, you will see I think the spontaneous generation of biogenesis, it's long before Louis Pasteur. Okay, so before Louis Pasteur generation, people were thinking about what? About if your beef soup goes bad, so what happens in the beef soup? It's spontaneously happened, or there's something in there caused the spoilage. So it's hard to imagine that we have to talk about this idea, but 100 years ago, 200 years ago, people are not sure whether the bad beef soup is due to spontaneous, there's something just happened. It just happens, so it's spontaneously. And then until Louis Pasteur, they found the microbes, different microbes will result in different characteristics of the wine. Then people start to realize the bad beef soup may be due to bacterial contamination. Okay, so that's called biogenesis. It goes bad because of one thing, not the beef soup itself. It's because the microbes come to the beef soup, so the beef soup will go bad. Okay, so that's the original idea. And also there's, for the past, we know the canning preservation already found in 1819. And so we can preserve the beans from canning. However, you see from 1895, it was found the first case that the green beans was in the can preservation. However, it still caused the contamination. And then scientists found that's because of the thermophilic spore forms in the can. Okay, so once you have the good condition, then the spore will start to ferment. So that's caused the sporulation of the green beans. And the pasteurization is through the Louis Pasteur. So that's the past. So what is the current status? We see there's some identification and characterization of new microorganisms. And there's an idea about predictive microbiology and the food shelf life. Okay, and also there's some environmental stress and microorganisms resistance. This part I didn't show any data. Actually, I have the data, but I didn't show it here. But we know there's a lot of so-called antibiotics resistant strains. Okay, so when we do some microbial isolation from fresh cut fruit and vegetables, sometimes we do see some bacteria will become antibiotics resistance, even with the same, for example, Pseudomonas. We probably isolate 20 Pseudomonas, but we will find there are 2 Pseudomonas strain may be antibiotics resistance. So, but why, we don't know. And we know there's a lot of resistant bacteria in the, of course, some of the bacteria have antibacterial resistance is because of they have the genes naturally. But some of, like Pseudomonas, it should be antibiotics or ampicillin negative, but while we tested our isolated strain, it may become ampicillin positive. Okay. Okay, so I will have another example. This is the research that we collaborate with one of the soya milk company in Taiwan. And also we did some tofu study as well in Taiwan. That's the idea that we want to know whether the microbial contamination in the, actually it's not the focus on the contamination. Actually, we are trying to understand the predictive microbiology. And I will explain that later on. So when you can think about that, why we are interested in this project, because when we, very often when we buy milk or buy soya milk, and they have the shelf life about 2 weeks, especially for milk. It's only 2 weeks in Taiwan. But actually, I don't know whether you have tried. If you have the milk in your fridge over 2 weeks, it's still okay. sometimes, but sometimes it will go bad. But if you are not, you don't open the package, sometimes, for most of time, it can prolong the shelf life until like 3 weeks or 4 weeks. Okay, so that's our initial idea, to see how safe these sterilization processing, and whether we can extend the shelf life for this milk or soya milk. In that case, we can prolong the shelf life, and we can also reduce the waste. Okay, we can save the food. Okay, so that's the idea, that we trying to do the microbial contamination in soya milk. And actually, this is a quite big company, and they do have a really good HACCP and all the regulation. So actually, we just want to claim that the product is totally safe. However, in one of our accelerated experiments, which means for the unpacked milk or soya milk, normally we will store it at 5 degrees in fridge, right? But in our experiment, we will tested the package, the soya milk, and we get the soya milk and put it into 5, 15, 12, 25, and 35 degrees Celsius, trying to accelerate their food product. And we did a lot of experiments. Actually, there's one batch of the experiment, we see the contamination with the pink colony. It's only pink, there's no other cells. So we're interested initially why this pink colony can survive in this soya milk. And because almost at the same time, the company told us there's several customers call to their call center and said they found their soya milk become pink. So it just accidentally, we found it at the same time. Okay, so we trying to identify what is this cells, and that's why let's see. For those pink cells, we isolated, and we're trying to identify. Of course, we use 16SR DNA sequence, and we do the DNA sequence analysis and trying to compare what is the gene sequence according to this 16SR DNA. And we found it's all Rhodococcus. And this strain hasn't been found in soya milk. Okay, so we think that's an interesting, this is the detail to see the map. Actually, we have 2 types of cell. One is labeled with DT. We have 4 strands of DT, and then we have a lot of RD somewhere, sorry. It's all labeled with DT R0 to R5. So we have a lot of similar colonies to do the sequence. And then we, because these strands were isolated from the soya milk, so for the predictive microbiology, the idea of predictive microbiology is the soya milk actually, it's, the characteristics of soya milk is because their pH is around 7. So between 5 and 7. So it's a relative high pH value. So it's not a good, it's very easy to contaminate it. And the second reason is because the soya milk with high protein. So it's very nutritious medium. So if there's any specific microbiomes in the soya milk, then according to predictive microbiology, you should be able to predict if you have 10 cells at the original product, how long those 10 cells will reach to 1,000,000 cells. And then that will cause the milk become bad. Do you know my idea? So that's why we called it predictive microbiology. So the predictive, the idea is actually originated from US FDA. They started to look at some, because in USA you will see every year they have like chicken, they have fresh salad, they have egg contaminated with salmonella or other strains. So they are curious about whether particular microbes prefer to stay in particular environment. So that's the idea. So if Rhodococcus is particularly like to stay in the soya products, then that will become a problem. So if you know how they grow in these soya milk products, then you can predict the number of the cells in the soya milk if you put this milk in a different temperature. So that's why we called it predictive microbiology. Okay. So in that case, we did the Rhodococcus in the soya milk. So we isolate the Rhodococcus, and we're trying to, actually we did two different things. Is we inoculate, we grow the, which means we grow the Rhodococcus back to soya milk, or back to the lab. We usually use TSB medium in the lab. So we put this, we inoculate the strands in the soya milk medium or in the TSB material medium. And we actually the experiment is not too difficult, but it time consuming. So you do different temperature from, actually we started from 5, 15, 20, 25, and 35. And you will see that's different time. Actually, for these experiments, we put it really long time. And interestingly, this Rhodococcus, if you are in the 5 degrees Celsius, they cannot grow at all. So it won't become an issue in the fridge. If you put the soya milk in a fridge and control the temperature well, then it won't grow. Okay. They even, you see that we put 10 A's, 100 million cells, and you see they remain, we call it inert. They do nothing there, okay? But they won't grow. However, if you started from 10, 15, 25, and 20, 25, and 30, and you will see they start growing really fast, okay? So this is the first predictive model. That model is how long they can grow in your medium and the time with the number, with the cell number, cell count number. So it's really easy for the first level of predictive model. And the second levels of predictive model is you converted the cell number into the growth rate. So you still have the different temperature, but you have different growth rate. In this case, we'll see that's the SM is soya milk. This is the TSB data. That's medium from our lab, and this is the soya milk. So from here, you will see that it seems that Rhodococcus is preferred to be in the soya milk, and compared to the TSB milk, TSB medium, slightly. Okay, so that tells you that this protein may like a protein-rich environment. These cells may prefer to stay in the protein-rich environment. Okay, so this is the second levels model. However, we didn't do the third level of model. What is the third level of model? Actually, for the predictive microbiology is if you can know the data from the industry, for example, the cold chain storage, the cold chain transportation, and through the cold chain transportation, if you cannot maintain a good temperature. So for example, we have a news that from Taiwan many years ago, like if you are in the summer in Taiwan and you can measure the temperature of a cold chain truck from Keelung down to Pingtung, then they may have 5 Celsius degree differences in the cold chain. So if you can get the fluctuated temperature, then you can apply you can also start repeat the experiment at different time and different temperature program. Then you will be able to predict if the product from the company to the customer through these transportation or processing steps, if they are not in a good temperature maintenance, then if there's a possibility contaminated by this strain, then how long you will see the microbial growth in your products. So that's the idea of the predictive microbiology. Okay, but for us, we don't have the permission to follow the industry track to go through so that's why we stopped here. But we did found this result, and because the company actually, as I said, we actually put from the previous data, we incubate the cells for quite a long time. So, and because they are UHT product, so we don't think that's the contamination from the processing. But we do think that's through the discussion with the company, we think that the colony, the bacteria may be contaminated by one batch of their packaging. Okay, so because they didn't sterilize their packaging, they didn't sterilize their packaging. So one of the possibility is from the packaging. Okay. Okay, so that's my first part, and because I don't think we can finish all of them anyway. So let's go for the next. I think we finished the past, and then we can have 5 minutes break. And for foodborne disease, in 1820s, I think the first happened in Germantown that has the sausage poisoning, and it was found it may be related to the botulism. And there's Vibrio cholera, as we said, that was identified by Dr. Koch. And then we started to know the cholera's contamination. And there's also in 1856, the Salmonella typhi from water contaminated with feces. And this was resolved by the chlorinated water. And the chlorinated water will reduce the number of Salmonella typhi, so that can reduce the disease, the chances of the disease. And in 1885, it's the E. coli and the infant diarrhea. Okay, it has been identified. And then we know that in more closely in 1984, there's Staphylococcus and infected beef that will cause the human death. And in 1895, there's Clostridium botulism and botulism from the contaminated meat. And these we already known like 100 years ago. And these microbes, we call it pathogenic microbes, that will cause illness, but generally do not affect the taste, smell, and appearance of the food. And that's why, once it happened, it's normally cause illness. Because before you eat it, you probably don't feel anything about these pathogenic microbes. And by the way, just remind you again that we said foodborne pathogens for the rotavirus, it's not foodborne pathogens because it's still pathogens, but because it cannot survive in the food, it cannot proliferate in the food matrix, okay? But it still causes the foodborne disease. So the primary source of the microorganism in food, according to our previous study, you can see that raw material is one thing, and acquired during production or harvest and handling, processing, storage, etc. All these processing will cause the microbiome in the food. And they may also survive in any preservation, treatment, and storage. And I think we want to focus on that yes, I will have the tofu story later on. So that's the raw material stuff. And for the treatment surviving any preservation, treatment, and storage, such as the new technique, we called it hydrostatic pressure, HPP. And the HPP, we know it's a new technique to kill the bacteria. However, is there any, they must have some cells can survive from the HPP treatment. And whether those HPP treatment, whether those survivors can still survive in the next circulation or next batches of HPP treatment. Actually, I think that's quite interesting for the new technology and the microbial contamination problems. Okay, so I will go through this really quickly. That I think microbial foodborne disease in Taiwan, and I think the rest of them for other. We still have some time. Okay, so we know that food poisoning or intoxication, there's two conditions. And if ingestion of toxins produced by microorganisms in food, especially such as food botulism, Clostridium, or Staphylococcus poisoning, food poisoning. Okay, and food infection means consumption of food containing pathogen organisms. Some pathogens can grow on food, just use food as a vehicle. And these we call it food infection. And some can grow on food. So the point is there's food poisoning or food intoxication and food infection. The infection means there's microbes in the food. But food poisoning means they may not have the microbes in the food, however, they have toxins in the food. Okay, so there are different types. So this is just the idea that the contamination happens in these places in Taiwan. And for last, actually, at least we have 30 years statistics showing that mostly it happens in the restaurant every year is the for most cases. However, so each cases may have several patients, and the most patients were from school. So, for example, the school lunch box from school. So the school is one of the place that has the most cases and most affected numbers of persons. And then the second, the first is restaurant, the second is the school, then the third is the, it happened at home. So for safety at home is really important as well. And this is the statistics for the microbial contamination. And this is the, I know you may not familiar with the English. This is the Vibrio parahamilocus. Okay, so we call it 肠炎弧菌. And it happens the most in Taiwan if we don't count the rotavirus infection. Okay, so for United States, as far as I know, and Taiwan now, if we count it in the rotavirus, and the cases, and the most cases will be rotavirus now. Okay, but if we don't count into the rotavirus, then in Taiwan it's the Vibrio parahemolysis. Okay, hemolyticus. Okay, so that's the 肠炎弧菌. And then in Taiwan we have the Staphylococcus and Bacillus cereus, and those three are the most cases in Taiwan. And this microbial contamination may be very different from different countries. But in Taiwan, because we have a lot of fisheries and we eat a lot of fish, so especially in summer, these clams and fish and this ocean product may cause the most contamination. Okay. And in the, interestingly, the types of contaminated products, I think we need to pay attention to this. This is the lunchbox produced by chain store or by supermarket, etc. So you know this lunchbox actually cost significantly increased in recent years, and it become very high numbers of cases. And the second part would be the fish products. Okay, for the past, it may be the fish product is the most affected categories, but now we switch to the lunchbox. Okay, and what is the development about the food microbiology, especially for the foodborne disease? And we can see that for mostly in Taiwan, the foodborne disease will be focused on the fast detection and effectively control of pathogenic microorganisms, especially the fast detection. However, there are not many research labs doing this fast detection research. And in foreign countries, especially in United States and in Europe, as far as I know, there are a lot of, it calls a lot of attention for pathogenic mechanisms, especially the biofilm formation and the pathogenic bacterial. Okay, so you will see there's a lot of conference about pathogenic related to pathogenic mechanism. And so these two are actually the biofilms and pathogenic microorganisms related each other. And also there's a pathogenic mechanism related to the environmental stress and the microorganism resistance, as I said previously. Okay, so this is our tofu study. Again, from soya milk, and we did some tofu. And the tofu story, we're trying to isolate it, I would like to explain the situation because the tofu is it causes a lot of problem in Taiwan. Although we have several companies, they produce really good tofu. However, in our traditional supermarket, we still have a lot of tofu without package, without any packagings. So in that case, the tofu may be exposed to the high temperature and without any protection. So in our case, we actually also collaborate with the company, and we're trying to understand the tofu from their product, whether how the quality is and what kind of bacteria that we can isolate it from the tofu. And interestingly, through the similar study as I show you the soya milk, we isolated the 2 bacteria which are form the major microbes in the tofu product. And one of them is called Bacillus. Okay, but we didn't we want to show you how we identify, because in the previous slide we want to do the detection. And the other cells, the other bacteria we identify is the Penibacillus. And the Penibacillus actually, it originated from Bacillus. And through the technique developed, now we can distinguish Penibacillus and Bacillus, okay? But they have really similar characteristics. So from our experiment, we isolate the bacteria from tofu, and we do the 16SR DNA sequence, and we also doing multi-TOF study. And multi-TOF is trying to understand the purified cells. They will determine, the multi-TOF would determine the proteomics in the identified cells, and then compare with their profile and see which cells they will belong to. And from these studies that we found the tofu product may be Bacillus cereus or Bacillus thuringiensis. So we know Bacillus cereus is one of the pathogens. However, Bacillus thuringiensis is the 苏力氏菌, so that's for kill the some of insects. Okay, so it's kind of it can be used to as a fertilizer, or it can inoculate or spread into the soil that can prevent the insects to invade this particular plant. Okay, so we are thinking, because these 2 are bacillus, so it's really hard to distinguish whether it's cereus or thuringiensis. So we did one thing is from literature search, we trying to find some of the specific genes from thuringiensis. So from the research, we know there are GYRB and NHEA, HDBLD, and Cyk, and CES, and CREO. And these are the, those 1, 2, 3, 4, 5 are the same genes that, these 5 genes, 1, 2, 3, 4, 5, 6, okay. So these 6 genes that showing, they are specific to Bacillus thuringiensis, okay. So we did, we isolate our strains, and we're trying to identify whether we have the number 1. The 1 here is the Bacillus from our tofu product. 2 is the Bacillus from our tofu product as well. And the 3 is the Bacillus thuringiensis. We bought the strain from our BCRC center. And then we have blank test. So we did the PCR test trying to identify the genes, whether they are series or they are syringes. And you can see from this particular genes, some of the genes, they will show up in both bacillus. Okay. So that's very often that we can see that. However, there's a paper reported that CRY1 is the real gene that will be happen, and that's important to syringes, but not series. So we tested the CRY1 gene, and we get the DNA from Bacillus cereus, and we also get the DNA from Bacillus thuringiens. And in this case, we did the PCR for the CRY1 gene, and then we found that it shows in our syringin control, but not correctly in our Bacillus cereus. So by this PCR technique, and also from 16 srDNA and multi-TOF data, we are sure that our strain is Bacillus cereus, but not Bacillus thuringiens. Okay. And this is another interesting, the same tofu, and then we get it from the company, and we're trying to identify, we want to see what kind of, whether, because previous study, we get the bacillus through the selection, through the identification. And however, we don't know whether that's the major Bacillus cereus in the tofu product. Therefore, we try that experiment again. We get the tofu product from the company. And once we get the company, the product from company, we do it at the 15-degree storage. At the first sample we collected is from the day that they give to us. And then we live in the 15 degrees Celsius for 2 days, 4 days, and 7 days. And we just collect all the tofu, and we use the blender to to homogenize the tofu and trying to extract the DNA from the tofu, from the whole product. We didn't do any, we just open the product and then collect all the samples. And then we're trying to see what is the microbiota in that product. So this is the data that shows the circle means we also, at the same time, we have another package that we measure in bacterial concentration, the numbers of bacteria. So from day 0, once the product arrived in our lab, we get about less than 10 cells. It's a log CFU only just about 1. So it's pretty good product. So at that time, you will see the cell number is only lower than log 1. And then it becomes log close to, and after 4 days, it reached to log almost 7. So that means there's a lot of bacterial growth. And then it dropped a little bit to log 6. And then we also did the microbiota analysis. You can see very interestingly in tofu. We see that from day 0 and day 2, where that mostly will be the original bacteria in the tofu. And we see a lot of portion, about 30% are lactobacillus, lactococcus. And then we see the geobacillus, and we see Sphingomyces, etc., and Weissella. Weissella is the strains, very interestingly, that very often found in stinky tofu, 臭豆腐. Okay. And we have other strains in this, and leuconostic. These leuconostic and lactococcus and Weissella, they're all fermented strains. Those strains are very often to be found in fermentation environment. Okay. And however, at 4 days and 7 days, we see that the red one actually is the Pennibacillus. Okay. So as we found that Pennibacillus and Bacillus, we see that from day 4, we found a lot of Pennibacillus. And from day 7, we see the Bacillus and Pennibacillus. So the microbiota can help us to understand the whole microbial population in the product. Okay. And in this case, we know that they can assure that our isolation from the Bacillus and Pennibacillus may be correct. Okay. So, and we also, as we said, the core study, core regulation at the beginning, beginning, we said he proposed that if you want to say one specific bacteria is responsible for the contamination or for the food poisoning or food contamination, then you have to isolate the specific strain and then inoculate back to the same product and see whether you get the same phenotypes of the product. Okay, so that's the basic idea. So we get this, we isolated these Bacillus and Pennibacillus cells. Then we put this, and we also, at the same time, we also isolate another R01 strains. So we put this strain, inoculate this strain back to soya milk because it's hard to do the tofu. So we put this bacteria back to the soya milk and see whether they can grow. And also we put back to the soya milk, and then we cook the soya milk. We heat the soya milk by the condition of pasteurization. And through the pasteurization condition, so that's after heating, we'll see that before heating, we know there's lots of cells, the Bacillus cells in the tofu. And after heating, you will see the Bacillus and Pennibacillus can stay in the medium. However, the R01, they were all killed. Okay, which means these Bacillus and Pennibacillus can survive through the heating process. That's why we think we can isolate these strains from tofu, because the tofu, they have very mild sterilization, pasteurization, because you can have high temperature, really high temperature to sterilize the tofu. So they normally, in the process, they have only low temperature sterilization. Okay, so in that case, you will see we still have the survival about 1.5%, the surviving rate, okay, and 0.9%. And so where are they from? Because we know Bacillus, they can form spores. So although we haven't tried whether we get those after heating, can we get these spores from the under microscope? But we know there's some endospores formed by Bacillus. So the Bacillus, once you heat it, as the kind of resistance to the heat. If you cannot, you don't have the good condition to kill all of them, then they may become, they form the endospore. So this is actually the Bacillus anthrax, not Bacillus cereus. But the Bacillus strain, we all know that can form endospore while they are encounter some stress, or they are lack of nutrients, then they will become endospore. So you will see the endospore, how the endospore forms actually when they were challenged by the environmental stress. They can duplicate their DNA, and then they will use their new DNA instead of their old DNA. So you see, they replicate their DNA, and then the new DNA will be moved to the one side of the cell. And they will form the plasma membrane around the cells. And you will see, and next step, they will start to form the plasma membrane in order to protect the newly synthesized DNA. And the old DNA then will be degraded. Okay, so once the old DNA was degraded, then the newly synthesized endospore will form another membrane. So actually, they will have two membranes inside. And once they form two membranes, then they will start to form the peptide glycan, their cell wall, and outside of the double membrane. And then once they form the peptide glycan, they will form another layer of spore coat. So you will see the endospore actually was protected really well. They have two membranes, and then they will have their cell wall peptide glycans, and then they will have another two membranes, and then they will have coated outside of those membranes. So actually, they have layers and layers and layers to protect the endospore. So once, normally it will, some studies showing that if you start to heat it, and then that will start to germinate it. So the heating is one of the activation to let them activate. So finally, the endospore can release from the cells. So from our previous study, we believe that there's endospore formed in our processing, and then so we can measure the bacillus afterward. Okay, so the last part will be the current status of the food safety, and we know that HACCP, as HACCP, has been established in most of the companies with good quality. It is required. Okay, so the novel technique for food processing, I think that's another issue that we need to understand, whether there's a lot of unknown in this process. And that's the interesting part also, the microbiology and the instant food. That's why we also did some fresh cut fruit, because the not only fresh cut, instant food, so for example, instant noodle or instant, a lot of new fruit or a new style of food. And that has been concerned about their food safety issue. So for example, we are now trying the fresh cut fruit. We also found actually the fresh fruit may contain quite high levels of microbes in the product. However, the product may not be, the spoilage or the one with cause the toxin or food safety issue, but it may affect the food quality and food flavor and the texture and the color. Okay, so I think that's the because now I remember there's about I think more than 1/3 of people in Taiwan, in average, they were eating out every day. If you added up the breakfast, lunch, and dinner. So actually, quite a large proportion of people, they are eating out every day. So that's why this instant food and fresh cut fruit and vegetables are needs to be concerned. Okay, so microorganism control from farm to table and the food safety regulation. So I think you can just get the idea from the slides that, as we said, that the tofu we found the bacillus, it might be from, actually we tested the bacillus series, maybe from their original soybean. Okay, so these are the tough way to get rid of these bacillus, because even they are from their raw material, but they can stay, they can form the endospore. So that's why in the product, it's possible they can stay in the product for a long time. And also the water, the air, and the packaging, as we see in soya milk. And of course, human, animal, and plants. And the fertilizer or insect, the compounds that the microbiome that uses to kill the insects, and from sewage equipment and stencils and product to product as well. Okay, this is another interesting idea to identify the contamination, where the contamination from. And this is, I forgot, it's MIT's study in MIT. They did in the United States. They did this study by tracking, or they explore the origin or the contaminations of microbes in an industrial environment. So they work with an industry company as well. But that company will focus on fermentation, especially for the cabbage. The cabbage, 泡菜. They're doing 泡菜. Kimchi, yeah. So they did not describe the detail for how they started this study, but they are trying to identify, because even for kimchi or other fermented fruit or vegetables, you do see contaminations. But where does the contamination from is always a difficult question. So they made a really careful plan for the study. And what did they do? This is the plant, the places, okay, the company. So according to their company, they follow the company's 规划，他们工厂的 factory layout. Okay, they according to this layout, and they you see that they coming from, this is the main water house, okay, and this is the fermentation room, this is the dry storage, and this is the packaging and processing room, okay. And they also draw out, like they did a lot of places. For example, this is the word is too small. And they also do this is the knives that they process the food. And this is the cabbage. So they have different cabbage. And interestingly, they not only test the microbes from the knife, they tested the microbes from the outer layers of the vegetable. And so they are leafy vegetables, so they take out leaves from the outmost leaves, and then to the inner part. So they analyze, they trying to understand the contamination is from the outer leaves or the inner part, or from the knife, or from which places, working places that makes the contamination. Okay, so they also did the, you see the door handles is the blue one. Okay, and the red one is vegetable handling. That's the places that they handle the vegetable. and the raw material, and the bathroom is here, and the dry storage is the blue one, and the light blue is the refrigerator. So that's the plan. Okay, and that's the fermentation. That's the barrels, I believe, the green one, processing room, and this is the fermenting barrels, because they're doing. They have the barrel to ferment. They put their cabbage in there, so they take the sample from everywhere, from the company. They also have the sink, the cutting board, and vegetable shader, and hand, and the mop sink, and the wash sink, all the sink, and the floor drain. Okay, so this is only one of the data. I didn't put all the data in here. You will see that relative abundance of bacteriophila across all surface. You will see that the bright color, bright red, means with higher abundance, and with lower color means low abundance. Okay, so from here you see the proteobacteria, these families of bacteria. You see they are from where? From the green one is the raw vegetable. Okay, and also with some red one. The red one is vegetable handling. Okay, and with the blue one is the storage place. Okay, and the pink, orange one, fermentation room, is not so many. But there's another type of Firmicus. They are from the blue one, dark blue, is door handles. Okay, and there's Actinobacter. It's may related to their fermentation. So you see that is from the processing room floor. Okay, so although they did not show any specific strain contaminated the food, however, they can see the overall, they can have the overall idea where they can see the bacteria from. Okay, so I think, to me, I think that's a very good example to extend the microbiota study to the industry, and to isolate or to see whether they should improve their hygiene or any way to control their bacterial contamination. Okay. Okay, so I think it's about the time, and the rest of the slides I will just quickly go through, because I know there's a professor from Japan. They will talk about this part as the factors affecting microbe growth. We all know the food composition, such as the tofu, soya milk with the higher pH, was easy to and more vulnerable to the contamination. And on the other way is the acidity with the low pH levels, then that can against the cell growth, bacterial growth. And there's a danger zone about the bacterial growth, so that's between 5 to 60. And sometimes you see some papers say 7 or 8 to 60 degrees, so that's called the danger zone, okay? Which means the bacteria is easy to grow during this period of time, period of temperature. And the time and the oxygen, whether that's aerobic or anaerobic cell growth, you have to be aware. If you are doing a microbiome experiment, you need to know the characteristic of your microbes, whether they belong to anaerobic or aerobic bacteria, and also the moisture. And so this is the pH. You will see that for lower part, of course, it's less chances to be contaminated. But with the higher, of course, if you are have alkaline pH, then it won't contaminate it, less chance to contaminate it. But you will see from here, from yellow to the the light green, the neutral pH, that would be the category that will easy to be contaminated by microbes. And I think this just the supporting data for you to see that the pH requirement for each bacteria. Of course, we'll say that all these bacteria has really wide range of condition that can survive from the environment. Not necessarily that its optimum is 7.2, but it can still survive at 5.5 or even 9. Okay, it just need to be aware. And this is the temperature, as we mentioned previous. It's the same that the different bacteria, but these data are from USDA, not from Taiwan, okay? So, for example, we seldom have Campylobacter contamination in Taiwan, and Listeria monocytogenes, we don't have that very often in Taiwan's case. So the growth with 32 Celsius to 45. Okay, so that's why when we do the test, so you need to test a wide ranges of temperature. And also water activity is a key condition. That's why when you do found that, for example, the rice with the cooked rice always have much higher chances to get the contamination rate compared to the dry one. Okay, and for the bacteria, yeast, and molds, of course, you can see the fungus, they can tolerate much higher range of condition, but bacteria have much lower range. Okay, so then I list some of the high-risk foods that you can think about it with the neutral in acidity and high in starch or protein. For example, our starch or our tofu, or high in moisture, those are belong to high-risk food. And with low-risk foods, you will see the high in acidity and high in salt or sugar or dry food and canned or vacuum packaged. These are the low-risk food. And finally, I think this natural microflora, raw fruits and vegetables, are usually non-pathogenic. However, if you are cultivate them or you make them in a domestication or in a particular food material, then that may become pathogenic or spoilage. Okay. So for the fresh cut fruit and fresh cut vegetable, probably is the worst case that we have seen. Yeah. So, but there's a lot of reasons, not only because of they are with high moisture and the storage condition, so that even they may be less nutrients, but they still can be contaminated by the processing. Okay, so how can we prevent these things? I think there's hurdle effects that will be continued by Professor Din's talk. I think she will talk about the hurdle effects. But with a simple exam that you can see, if you have the homemade jam, and there's a hurdle, by hurdle technique, you can treat with the heat, and then you add sugar. So that's double protection, and you can get the frozen storage, and then you can keep the jam safe. Okay. Okay, so finally, it's from farm to table. I don't have time to talk about this from farm to table. All these processing may have the risk for food contamination, especially that the transportation in United States, there has one example for ice cream contamination. That's because of the truck carry the egg first, carry eggs first, egg products first, and then they do the second round transportation with ice cream, and then the ice cream was contaminated with salmonella. That's from, finally they found that's from the egg product. Okay. Okay, I think that's all for me from today's talk. And if you have any questions, you're welcome to contact me. Okay. All right, any question then? If not, then we'll say bye to everyone. Okay? All right, bye.
Yes, we don't have any question. Thank you for today.
Okay, thank you.
Thank you. Goodbye.